Hello and welcome. I'm very pleased to introduce to you Professor Hans-Ulrich Childhouse from the Institute of Pathology at the University Hospital Essen and West German Cancer Center Essen in Germany. He's going to present to you today the fusion type dependent biomarkers impacting overall survival in synovial sarcoma that he revealed by HTG Transcriptome Profiling. This presentation is from the UK Biomarkers Congress in Manchester in 2021.
Thank you very much for this kind invitation to this meeting here. I'm Hans-Ulrich Childhouse. I'm a pathologist and molecular pathologist from Essen, Germany, and from the West German Cancer Center. And as you will see, I have a specific focus on sarcoma pathology as well and molecular diagnostics of sarcomas, and therefore I'm very happy to share some still preliminary insights in what we did with synovial sarcomas, a very interesting sarcoma subtype, and we have analyzed them with a novel and recently introduced transcriptome profiling approach. Here are my potential conflicts of interest. You can see personal things, institutional things, and I would like to stress that this study, which I'm going to present here, has been supported by HTG Molecular Diagnostics. A few words on synovial sarcomas and on the background of this. Synovial sarcoma is a group of soft tissue sarcomas which are usually clinically aggressive, and unfortunately it affects usually young patients, adolescents, young adults, the so-called AYA group of patients. So that means this makes this tumor a very interesting, very important field of research to improve diagnostics, to improve treatment options, etc. So beside this more general clinical relevance of this tumor, most synovial sarcomas are clinically aggressive in terms of local relapses, which can occur simultaneously or metachronously, and or distant metastases. And interestingly, there are clinical different subgroups which have been known for years by experienced clinicians. Some of the patients suffer very early from a very early relapse or very early distant metastasis, but there are others where the progressions occur very lately, sometimes even after 20 years of initial diagnosis. This is another interesting clinical information, and on a molecular level, this tumor is a paradigm for gene fusion-driven cancer. These synovial sarcomas have 100% of these tumors have these SS18 gene fusions, and 100% of SS18 fused tumors are synovial sarcoma. So this makes this very clear in terms of the underlying biologic event. So there are, however, at least two different types of gene fusions occurring there. One is the SS18 fusion with SSX1, and there's another group which accounts for roughly 1/3 of the overall cases, which is where SSX2 occurs instead of in SSX1. Very rarely, also SSX4 can occur as a translocation partner. So what do these chimeric fused proteins do in these tumors? And there has been substantially increase of our understanding of these tumors. We know that these SSX, SS18 gene fused or chimeric proteins interact with a SWI/SNP, the SWI/SNP complex, and these interact with this KDM2B, which is an histone demethylase, and this is part of the component of a gene repressive complex, the so-called PRC1.1 complex. And this whole interaction leads to an aberrant activation and aberrant expression activation of genes which are developmentally regulated and which are usually repressed in adult tissues. And this recapitulates an immature, more embryonic-like type of gene expression levels. So that means we can say this SS18, SSX1 fused or chimeric protein deregulates developmental programs by hijacking of the transcriptional repressive complex, the PRC1.1 complex. So this makes this a good candidate to be studied by gene expression analysis. So since we would expect that these synovial sarcomas would express a completely, more or less completely different repertoire of genes compared with other sarcomas, which are not driven by these specific SS18, SSX1 or 2 gene fusion. So these genes, or this fused gene, consists of a major part of this SSX or SS18 gene, which has been formerly known as SYT. This has been renamed to SS18 since it's located on chromosome 18. So that means the major part of this chimeric protein consists of this SS18 component, and there's only a smaller component which is contributed by either SSX1 or SSX2. And interestingly, both proteins SSX1 and 2 are both different chimeric. The chimeric proteins differ in only 13 amino acids. So that means there's a really tiny difference between those different versions of this gene fusion. So I think this makes it plausible that both translocation subtypes lead to the same tumor subtypes. However, it has been demonstrated by several groups that there are obviously, from a clinical perspective, also some differences between SSX1 and SSX2 fused synovial sarcomas. For example, the SSX2 rearranged tumors do show a little bit more predominance of female patients. On the other hand, these biphasic morphology, which is defined by a spindle cell and an epithelioid component, is underrepresented in the group of SSX2 tumors. Also in terms of the overall survival, both groups seem to differ a little bit. So as you can see here in these overall survival curves here, and this should or is obviously independent of the occurrence or non-occurrence of metastasis at the initial diagnosis. So, however, these findings have not been confirmed by other groups. However, our clinical experience and our clinical experience points toward that among those SSX2 patients, there's the higher likelihood to have these late responses and to have to survive longer. So that means this makes it interesting for us, and we wanted to define novel biomarkers which allow us to further study these differences between these tumor subtypes. As already mentioned, there's this change in this gene expression repression makes this tumor interesting in terms of gene expression analysis, and this has been demonstrated already or has been studied already in previous approaches. You can see here one example of studies which have been published based on cDNA microarrays, demonstrating that the group could demonstrating a nice difference between MFH, this is malignant fibrous histiocytoma, which stands for an undifferentiated pleomorphic sarcoma, and synovial sarcomas. I think this is not so much surprising. They also demonstrated some of those tumor, some of those genes which have already been demonstrated by others, for example, AXL. And they could demonstrate also nice difference between biphasic and monophasic tumors. And this is also not so much surprising, since this is mainly driven by keratins. And these keratins are those proteins which make these epithelioid morphology in these tumors. However, the authors state they could not demonstrate any difference between SX1 and SX2 gene fusions, and they state here this negative finding may be unexpected, given the known correlation of SYT fusion subtype with survival and areas of glandular epithelial differentiation, but the study group and the technical platform may have been inadequate to detect the likely much smaller difference in gene expression. So this makes it interesting for novel developments, for also in novel technologies. So, so what we aim to do here is we wanted to explore how comprehensive gene expression analysis can help to define synovial sarcoma subgroups, and we wanted to determine how synovial sarcomas as a group differ from other sarcomas based on the entire picture, comprehensive picture, and we have chosen two different comparison groups. One is in sarcomas with other types of gene fusions, what we call FAS sarcomas here in this study, and we wanted to compare these with the morphologic mimickers, other spindle cell sarcomas without any known genetic alterations. And we wanted also to identify differentially expressed genes between SSX1 and SSX2 subtypes of synovial sarcomas. What did we do? We did a retrospective analysis of archival material. The paraffin blocks were up to more than 4 years old. They have been stored under standard conditions at room temperature. No specific tissue. We have used resection specimens and biopsy specimens, with the majority of samples representing resection specimens. We have included 45 synovial sarcoma samples with known fusion status, and the comparison sarcomas 23 spindle cell sarcomas, NOS, and 8 fusion positive sarcomas other than SS18. So the technology what we have used was the HTG Transcriptome panel, which is more or less based on this nuclease protection assay procedure methodology, which is basically a method to determine, just to count copies of RNA in tissue without any extraction and without larger amplification steps. So basically, the target RNA is covered by probes which are containing these wings and wingman structures, and this and the entire lysate is digested by an S1 nuclease, which digested anything of nucleic acids but keeps those DNA, RNA chimeric structures there, and this is how they are kept there in these lysates, and then you can easily adapt ligate adapters there, and can, with a few amplification steps, you can just count the amount of these numerically these molecules. This panel, what we have chosen, contains roughly 20,000 probes, including controls, and we used 1 or 2 sections per block, only 1 section. So this is a nearly fully automatized workflow. The majority of things are done on this HTG device here. And then we have finally used an Illumina NextSeq 550 sequencer for reading on counting the number of molecules there. This platform has some advantages over RNA-seq, especially this simplified workflow. This is really something what we appreciate with this technology, and has a high pass rate, as I will demonstrate. And this is higher than on RNA-seq from FFPE material. This panel comes also with some QC metrics, which we have also used in our cohort, which measure the RNA quality, the read depth quality, the background signal quality, and the DNA suggestion quality. And this is what we have seen here in our from our cohort. We can demonstrate that based on the RNA quality, we can see that we have in all of those 76 samples, we have all samples in place. There was no issue with the quality. Here, in terms of the read depth, also all tissues met the criteria of quality, and also in terms of the background signal, we could accept all of those. So this was very good to see that also these archival materials contain a lot of high quality or reveal a high quality of samples. There were only 2 samples which we excluded based on incomplete DNA digestion. But these were only 2 out of these 76 samples. Based on a pre-specified probe set, we could demonstrate that we could nicely separate these different groups of sarcomas from each other. We could nicely demonstrate that the synovial sarcomas cluster quite close to each other. However, this wasn't, from my perspective, a moderate separation. These pre-specified probes that included also SS18, for example, or other genes which have been published in the literature. If you look at the entire picture and use more than 10,000 genes simultaneously in this principal component analysis, we can nicely see that we can separate the spindle cell sarcomas from other fusion-positive sarcomas and from synovial sarcomas. Interestingly, there are some cases which are quite more similar to each other, and these are currently interesting cases which we are currently working on. And there was one outlier case which has been demonstrated a completely different gene expression profile based on a neoadjuvant treatment, which induced a lot of cell damage here as well. If we compare the spindle cell sarcomas versus the synovial sarcomas, we can exactly demonstrate what we expected, that there's a huge number of deregulated, differentially expressed genes there. More than 5,000 genes are differentially expressed, and this is exactly what we expected. And we can also look down to these different types of genes, and you can see as those what we expect. TLE1, for example, is dysregulated, etc., etc. So that means you can nicely identify also this gene of interest there. And if you go with the top 50 deregulated genes back to this principal component analysis, you can see close to perfect separation between these different tumor entities there. And this is a list of those genes which are deregulated between these genes. And again, TLE1, this is what we also use as a diagnostic marker, comes up here very well. But there are a lot of interesting genes what we are currently evaluating and validating with other methods. Also, in terms of SSX1 versus SSX2, we could find an interesting observation here. And yes, we did find differentially expressed genes between those groups, SSX1 versus SSX2. This is only 13 out of those more than 10,000 genes which have been investigated there. And This is very interesting to see, and this is also something which provides us new insights into novel biomarker assays and biomarker development to separate these groups. Among them, there's also histone deacetylase, as you can see here. So that means this tool enables us to identify those few genes which are making the difference between SSX1 and SSX2. So with this, I would like to sum up here. Synovial sarcomas are aggressive spindle cell sarcomas showing this consistent translocation, either with SSX1 or SSX2. And the overall survival is thought to be better in patients with SSX2 than in SSX1. So there is a need to further evaluate these changes, since this is not yet so much clear clinically. And we are in these broad extraction-free transcriptome profiling help to identify differentially expressed genes that are linked to the translocation type, which provide us a novel basis for developing biomarkers for these subgroups. These differential expression profiles between synovial sarcomas and other spindle cell sarcoma types were in line with the previous findings, and the identified biomarkers offer new opportunities for improved diagnostics for aggressive synovial sarcomas. With this, I would like to thank the people who were involved in this project. I would like to thank my team from Essen. I would also like to thank the HTG team, which supported us a lot with all of these technical things. Thank you for attention, and thank you for listening. And I'm open for any questions if there are some.